Good evening, everyone. I am thankful to Varshney Teachers Group and particularly Gagan Gupta sir, who by inviting me to this session have given me the opportunity to think, discuss and deliberate on the most important topic, assessment reforms against the backdrop of National Education Policy 2020. The policy drafted under the chairmanship of former ISRO chief Shri K. Kasturi Rangan was approved by the Union Cabinet on 29 July 2020. And it is only the third major revamp of the educational framework in India since independence and aims at making India a global knowledge superpower which would align the country's education system with the needs of the 21st century while remaining rooted to the Indian values. The policy offers an integrated and flexible approach to education and provides an educational roadmap for the country. Now, with the policy document already in the public domain since last 5 months, I believe we all are now aware about the reforms that have been proposed. So today I will be talking about the reforms related to assessment, which is the bridge between teaching and learning and is in fact more difficult than teaching. So now first let us give a quick look at the journey of evaluation system and how it has changed over the period of time, what the new policy has in store for us and what is the role of teachers, how do we implement it. Now during the Vedic period, Gurukul was a type of education system in ancient India. Evaluation in the Gurukul system was mainly done in the form of oral recitation, informal observation and practical skill training. Oral recitation, informal observation, and practical skill training. Later, educational sanghs and madrasas were set up, but the assessment was still done orally. And although India had a rich tradition of imparting knowledge, modern India failed to capitalize on its initial age due to years of colonial rule. And post-independence too, examination-based education system was introduced. But by 1950s-60s, need for separate education policy was felt to take the country ahead, and committees for the same were set up from time to time. On the basis of reports of various education commissions such as Medalier and Kothari Commission, the first NP came in 1968, which recommended for a shift in the focus of evaluation from mere certification to improvement in learning. The second policy came in 1986. It insisted on evaluation being the integral part of the teaching learning process and again de-emphasized memorization. Use of grades in place of marks and introduction of CC were some of the innovative measures suggested by the committee. The concept of CC was excellent, but the rules for assessment were not defined. It was misinterpreted as periodic tests and its implementation was pathetic. The journey of student assessment has thus gone a long way, passing through oral testing, written testing, public examination, internal and external assessment, CCE, and competency-based assessment. And over the time, India has moved from examination to evaluation and now to assessment. And it is our responsibility to see that NEP 2020 does not meet the same tragic end as previous policies. As rightly observed by Radhakrishnan Commission, if there is one thing to be reformed in our education, it is the examination system. The policy aims to end the rat race of marks and inculcate understanding about the success of failure. साइकोलॉजी के अनुसार मृत्यु इंसान का दूसरे नंबर का डर है, फेलियर पहले नंबर पर विराजमान है. लोग मरने से कम फेलियर से ज्यादा डरते हैं. Scoring not only good marks but full marks is the only aim of the student, because society places such students on a high pedestal, and even if a child has scored 92%, there is an atmosphere of mourning in the family. The student's ability is being assessed by just the pen paper test. Who is responsible for making exams a source of stress? Parents, teachers, community, society. Yes, we all are. And so who will have to find the way out? We all have to, in a joint and collaborative effort. यूं ही नहीं आती खूबसूरती रंगोली में, अलग अलग रंगों को एक होना पड़ता है. हम सबको भी एक होना पड़ेगा. We need to transform the culture of assessment. And to transform the culture of assessment, we need to transform ourselves. Not because we are not good, but to be better. Teachers have to be prepared for reformation in the assessment system by the year 22-23 academic session. So now we saw the history of examination. Now let me have a look at the major changes that have been suggested in the policy regarding assessments. From grade 9 to 12, we'll have just broad look on it and then we'll discuss about the assessments. So board exam as proposed by the policy will be redesigned to encourage holistic development and discourage coaching culture. These exams to be made easier in the sense that they test primarily core competencies rather than the content memorization. To reduce pressure and coaching culture, boards will think to reduce the curriculum and develop semester system. Students will be given to choose range of subjects in which they take board exams and not only a narrow band of single stream. They can cross streams to select the subjects of their choice. There will not be rigid separation of streams, no silos at all. All the students will be allowed to take board examinations on up to 2 occasions during a year. One main examination and one for improvement to eliminate the high stakes aspect of board exam. These exams will be renamed as improvement exams. Anything can happen on the day of the exam for a child, because of which he could not do well or miss the exams. So should he suffer for those unfortunate occurrences for the entire life? एक मौका तो बनता ही है. And new policy has taken care of this. For class 12, we already have 2 levels in maths, basic and standard. Policy mentions that all the subjects will have 2 levels, and this will be extended up to class 12. Examinations from grade 1 to 8. Instead of exams being held every academic year, school students will only attend 3 exams in class 3, 5, and 8. And census assessment will be done in these key stages to monitor the achievement of learning outcomes. The result of school examinations will be used only for developmental purposes and for continuous monitoring and improvement of the schooling system, not for certifying the capability of the students. There is also provision in NEP regarding support for gifted students. Talented students will be encouraged to pursue realism beyond the general school curriculum. If a class 5 child is capable of appearing graduation exam, he'll be allowed to do so without much hindrance. Presently, the parents have to run from pillar to post for getting permission. It's not easily granted. So this policy has a provision which will make it easier for everyone. The next most interesting thing is report cards for the school-based assessments to be redesigned to make them holistic. They have been made 360-degree multi-dimensional report by including self-assessment and assessment by teachers, parents, and peers. It will include progress of the child in inquiry-based learning, quizzes, role play, group work. Thus, the report cards to reflect the progress and uniqueness of learner in the cognitive, affective, socio-emotional, and psychomotor domains. Artificial intelligence-based software will be used by students to track their growth through school years and help students make optimal career choices. And coming to feedback. Comments by teachers are very important in the life of a student because they stay inscribed not only on the report card, but also in the minds of students. And yet, teachers have been found to use it so casually. Many times, remarks on report cards contain generalized statements chosen randomly from the list of 20-30 statements. NEP says the feedback on the report card need not be only a post-mortem on how good or bad the child had been, but it should be like a medical report on what needs to be his behavior in future. The feedback should be forward-looking and not backward-looking. Feedback should not be of the view through the rear view mirror, but it should be from the windscreen mirror. It is going to be very hard to do. But that is the stuff which is going to make the difference. Don't you think so? Now, these are the broad provisions in the policy regarding the assessment. Now, in order to implement the assessment reforms, let us first understand the assessments. इन्हें सिर्फ हम ऊपरी तौर पर नहीं, थोड़ी गहराई से समझें. Now, most of the audience, I believe, will be educators and otherwise also. I want to ask you one question just to feel more connected once again. You can write in your chat box. My question is, which type of assessment do you think is better? Summative or formative? Just write only the one word which you feel is correct. Summative assessments are better or formative assessment? I will find it in the chat box. Supriya Shinde is saying formative. Priyanka Varshney ma'am is saying both. Okay, Sunanda Ingle formative. Sania Kumari says formative. Rashmi says okay. Okay, yeah. For Prachi formative. Jayashri formative. Ravi Kumar formative. Formative, both. Deepa Vai says both. Okay. Different replies I'm getting. The feedback. Formative, yeah. Neha Singh, both. Lalit Gupta, both. Okay. Now, we know that there are three types of assessment. Assessment of learning, assessment for, and assessment as learning. Now, assessment of learning is the assessment that becomes public and results in statements or symbols or grades about how well students are learning. It often contributes to important decisions that will affect students' future. It is important then that the underlying logic and measurement of assessment of learning be credible and defensible. The purpose of this kind of assessment is usually summative and is mostly done at the end of a task or unit of work. So this is summative assessments are like stock taking of the inventory in education. We teach students and take stock of their learning at the year end. Thus, year-end exams, or for that matter, even periodic tests are assessment of learning, because it just gives you feedback of where you are on the academic ladder, but does not do anything about it. It is as good as, boarding a flight for Mumbai, and when you actually land, you are told by the pilot that, I'm sorry, you are at the Pune airport because of this reasons and route. But I can't do anything about it because I have to get other passengers. A teacher cannot be a pilot and at the year end just inform the students who summative assessment by writing comments about his incapabilities and shrug off the responsibility to take his students to the necessary destination. What the pilot could have done, or rather what he should do, somehow fight the rough weather, contact the ground staff, navigate his flight to take a different route, and ensure that passengers land at the Mumbai airport, the desired destination. Similarly, a teacher also has to be an efficient navigator and navigate the child through rough weather, or by taking different route and see that the child reaches the destination. That is, achieves the desired learning outcome. This intervention during the journey is what we call formative assessment. NEP says we should have more of formative assessments where the assessment is for learning and as learning. Formative assessment not only help teachers to identify learning needs and problems of the class, but in many cases, these assessments also help students develop a stronger standing of their own academic strengths and weaknesses. If the students know what they do well, what are the expectations from them, what they need to work harder on, it can help them take greater responsibility for their own learning and academic progress. So what is the simplest way to conduct formative assessment? It is done by asking questions during the lesson. Through formative assessments, we can not only assess the knowledge and understanding, but we can also assess application of knowledge. For example, when I ask, what is the conversion of water from water vapors to liquid state called? The answer is condensation. So this is a knowledge level question. And when I ask the child to identify the process of conversion of water in the formation of dew on leaves, is it evaporation, condensation, or freezing? Here the child uses his understanding to answer. Condensation. So this is an understanding level question. And when I ask, how can you incorporate processes of evaporation and condensation in distillation of water? This becomes an application level question. Similarly, in mathematics to knowledge, understanding and application level question should be included in formative assessment. For example, when I ask, what is the sum of measurements of a triangle? It is a question to assess the knowledge level. If I tell them to measure the sides of the given triangle and name it based on its side measurements, I am assessing their understanding. But when I ask, if two angles of a triangle are 65 degree and 35 degree, then what type of the triangle will it be? Then I am assessing the application of his knowledge. So we need to ask more and more application level questions and less of where only their factual knowledge is tested. For this, we need to focus on framing good questions. Now, my next question again to the audience is, what type of questions do you think are good? Closed or open-ended? We know different types of questions. I would like to see the chat box, your replies. What type of question do you think are good? Supriya Shinde says open-ended. Deepa Vahi ma'am says open-ended. Ravi Kaur open-ended. Sania Kumari open. Everybody says for open. BK Varshney sir says both. Good. Nimesh Singh open-ended. Okay. Shehzad Safi open-ended. Lalit Gupta says open. Anil Kumar Mishra open-ended. Neelam Kumari both. Bandit Saring both. Okay. So we are getting mixed replies. According to me, it is not always necessary to ask open-ended questions. You may ask open-ended or closed, but it should involve thinking by the child. Questions should not be asked just for engaging students. Don't give them hints, elicit the answers and move on. Ask questions to make them think. To ask such questions which will encourage thinking, teachers need to think. Rather than planning what a teacher will say in the class, more time needs to be spent by teachers on planning what type of question should be asked. It should involve higher order thinking, critical thinking, logical reasoning. Don't just ask close-ended questions which require only factual recall or algorithms which the child is already aware of. Ask questions which require higher order thinking and then give time to think, because higher order thinking requires time to think. At times, few questions are asked repeatedly in the form of revision. And one fine day assessment is done by again asking questions from the same bunch. At other times, the questions are just being bowled at the speed of rain and students are also batting the answers. But is there any learning taking place? Cognition doesn't work as fast. So such lessons should not be confused with learning taking place. Pace needs to slow down for learning. Teachers need not be airplanes, and classrooms is not an airport to just move in full speed from one end to another and then take off. It needs to have some hinges where you ask questions. Not great piece of work, but simple short questions, and decide whether you need to move on or redo the teaching in a different way that all the students understand. So while teaching, we need to have formative assessments for which questions should be planned, should be of higher order, and also after asking, just be silent for a few seconds to allow him to think. Poor thing, by the time the child understands the question and starts thinking, connects the points of the answer, the teacher has moved to another child. Now, one more question I would like to ask you. Now, what happens in your class when you ask questions? I'm also a teacher. I know what happens. So I want honest answer. I want to see what happens in your class. Do they raise hand to answer question? Or do they scream and shout away the answer? There's a noise in the class. and you're scared the school head might be on the round and you'll be answerable for the chaos. I'm waiting for your answers. Pin drop silence. Supriya Shinde ma'am says, intelligent students only answer. Sunanda Ingle ma'am says, pin drop silence. Ramanik ma'am says, at times they leave if they don't know. Dr. Vinay Kumar, mixed, some raise hand. Ravi Kaur, children who knows they start screaming. Jayashri Adav says, shout the answer. Yeah, so different type of responses. So how many of you allow the students to raise the hands and then ask answer from that child? You can send me an emoji of hand raise if you have. There's an emoji of hand raise. You can just insert the emoji of hand raise and I can find it. Otherwise, you can just say allow, not allow. If you're not able to find emoji, you can just write allow, not allow. My question is, how many of you allow the student to raise hand and then ask answer from that child? So allow. Sania Kumari says allow. Sunanda English says allow. Priyanka Varshney ma'am says allow. Okay, one of you have raised hand. Okay, allow, allow. Supriya Shinde ma'am says I do. Miss Chandra Satsan, they have expressions that show that they are thinking and they start raising their hands. They are excited. Very correct, Jayashri ma'am. I raise the hand and answer as well. Allow them. Dr. Vinay Kumar, Kalpana Varshney ma'am. Yes, yes. Okay, I'm not naming everybody. But yeah, there are mixed response again. I can understand asking question in the class of minimum 40 students. I know it's easier said than done because the students are after all children. When they know the answer, they get excited. They raise their hands to answer, make noise. Sometimes even before you complete the question, they will give away the answer. But smart teachers can have control on the class by not allowing anyone to raise the hand. Because when they raise the hand and you don't ask them, they get disheartened, bored, and demotivated. And if you ask them, and when required, explain them, correct them, so now these already smart children get smarter. And the ones who did not raise the hand, were not asked, remain there, and the gap between these two has increased now. So have the rule of no hand raise. Take the control of the class by informing that this child will answer and these others will assess whether the answer was correct. And if not, what was wrong? At which step the child is going wrong? Now everyone is going to be attentive. You've already created an environment where participation is obligatory. The class is in control. And this way, you are facilitating peer assessment, which the education policy is talking about. This intervention by the peers will not be disliked by the student. And while this child is thinking, the others wait patiently and listen carefully to what he is going to say because they have to assess him. In this way, a teacher can elicit evidence of learning from the students, not only from whom the question has been asked, but also others. And you actually save the time. Now, suppose the child says he doesn't know the answer. It happens. Still, the teacher should say, okay, I'll get back to you. And after asking few other children, she must go back to him and find whether he can answer now. Now, this time, instead of telling him to repeat the same answer, you can ask out of the 3 answers just which he heard that were given by the children, which one do you think is the best? Suppose the child still says he does not know the answer, the teacher can ask the child, okay, give the wrong answer and we will correct you. The student knows that he has to answer. There is no way out but to participate in the engagement. So the questioning technique should not be like a ping-pong tennis ball, initiate response type. It should be a basketball game where one asks the question, another gives the answer, some other confirms whether it is correct, and still another evaluates. In this way, teacher has to be creative and innovative while doing formative assessments. Another way to ensure attention of child while doing formative assessment is keep a box with paper strips, each containing the name of the child, and randomly pick the strip and ask the questions to the child whose name appears on it. Also keep the strip back into the box so that the child understands that there are chances that he is going to be asked the question again. So he needs to be attentive. Otherwise, once his chance is over, he'll stop listening and disturb the ongoing engagement. This way, the teacher has to keep them on toes, be in control of the class, and keep reinventing new ideas for a classroom. Each and every child must be engaged in the teaching learning process so that you get a responsive class. At the same time, it is not always necessary to ask questions as a part of assessment. For example, while teaching pollution to children, instead of asking direct question to name the most polluted city of India, and children just popping up with different names, Bhopal, Indore, West, or anything. Instead, I can just put a statement saying Delhi is the most polluted city in India. Sorry, Delhi people. What do you think? Now the student here is obliged to not just agree or disagree with the statement, he has to back up the answer with reasoning. So we as teachers need to improve our questioning technique to elicit desired answers and make necessary interventions wherever required. Educationist Dylan Williams rightly observed that as teachers, we need to be clear from where we are starting, that is by a short diagnosis test, be clear about destination, learning outcomes, plan the learning journey, that is lesson plan, questions to be asked, etc. Begin the task of learning, make regular checks along the way, make adjustments as conditions dictate, modify the teaching methodology and questions according to the students. And in this way, what we are doing is adaptive assessments, which has been mentioned in the new education policy. Some of you might feel it is very easy to talk about the methods of adjusting, but incredibly hard to do in practice. But then who said teacher's job is an easy job? And well, if we have chosen the toughest job, so let us face the music happily and creatively. NEP has not only emphasized peer assessment, but also self-assessment, which means activating students as resource learners for one another and also activating students as the owners of their own learning. Now let us come to some myths in teaching. One or two I'll take up, not more. Now it is a myth that if the teachers are strict, there will be student learning. Some of us think that if they will behave hard and the students will learn thoroughly. But the fact is that if students are not interested in learning, the learning is not going to take place, no matter how strict you try to be. Another extreme myth is a term which is popping very much these days, and it is that teachers have to be facilitators and students have to be the active learners. Teachers have to facilitate learning, and it is
Shobha ma'am.
Yeah?
I'm so sorry to intervene in between. Ma'am, there is a request from the participants if you can be bilingual.
Okay.
Thank you, ma'am. Thank you.
Acha. I can add Hindi, you mean to say?
Yes, ma'am. Yes, ma'am.
Okay, I didn't know that earlier. Doesn't matter. I'll try to be as far as I can. Now, another myth. ये गलतफहमी है हम लोगों को कि टीचर्स को फैसिलिटेटर्स होना है और स्टूडेंट्स को एक्टिव लर्नर होना है. टीचर्स को लर्निंग फैसिलिटेट करनी है, उनको मदद करनी है, और ये इट इज अप टू द स्टूडेंट्स. ये तो स्टूडेंट्स का काम है, उसको तो पढ़ाई करना है. मैंने तो भाई सबको पढ़ाया एक साथ और अभी स्टूडेंट के ऊपर है, और इसमें मेरी कोई रिस्पॉन्सिबिलिटी नहीं है. अगर बच्चा नहीं समझ पाता तो मैं क्या करूं? मैं तो 40 बच्चों को एक साथ पढ़ाया, वो समझा और ये क्यों नहीं समझा. I have even heard some of them saying that I have placed the food. मैंने तो खाना बच्चे के सामने रखा और ये बच्चे का काम है उसको लेना. मैं थोड़ी देर उसको खाना खिलाऊंगी, उसके सामने परोसा गया, वो खुद ले. But the real teaching is striking the right balance between these two extremes. हमें इन दोनों का बीच का रास्ता लेना है. ना तो हम ये कह के छोड़ दे कि हम फैसिलिटेटर्स हैं, बच्चा अपने आप सीखे, और ना ही इतना हम स्ट्रिक्ट बने कि हम अपना ही कानून चलाए. हमें बीच का रास्ता लेना होगा. एक एग्जांपल देते हूँ. हम सब कभी न कभी मम्मी रहे हैं, और जो मम्मियां नहीं है उन्होंने मम्मियों को खाना खिलाते देखा है. अगर बच्चा छोटा होता है और वो खाने को तैयार नहीं है तो हम उसको कोई झुंझुना दिखाते हैं, कुछ आवाज वाला खिलौना दिखाते हैं, ताकि किसी तरह बच्चे का ध्यान दूसरी तरफ आकर्षित करके उसे हम खिलाने की कोशिश करते हैं. कभी कुत्ता, बिल्ली, चिड़िया उड़, तोता उड़, मैना उड़ करके हम उसे फिर दूसरा कौर खिला देते हैं, मुंह में डाल देते हैं. कई मम्मी ऐसे करती है, हम सबने किया है। अब ये 21वीं सेंचुरी के बच्चे हैं, स्मार्ट है, बच्चे जानता है कि मम्मी ऐसे करती है, जब भी मम्मी मुझे कुछ दिखाती है, अगर मेरा मुंह आ रहा है क्या तो मम्मी ठूस देती है, बच्चा समझता है। तो वो क्या करता है, वो मुंह में एक और तो ले लेता है, पर ना तो उसे चबाता है ना उसे अंदर करता है। अब मम्मी करे तो क्या, तो मम्मी क्या करती है, पेशेंटली इसका इंतजार करती है कि वो खाना अंदर करे। और इस बीच अगर कहीं मम्मी थोड़ा सा, चलो बच्चा खा रहा है, जब तक एक काम करके आती है, काम निपटा के आती है, तो बच्चा क्या करता है, उसे थूक देता है। और ये प्रोसेस एक कौर पे चलता है, दूसरे कौर पे चलता है, तीसरे कौर पे चलता है, फिर एक दिन में 3 बार खाना, दिन महीने साल यही चलता रहता है। पर कोई माँ कभी अपने बच्चे को खाना खिलाना छोड़ती है क्या, थकती है क्या? नो, because though she was aware that being mother is the toughest role a woman has to play, she chose to be one. And this story is not ending here. कहानी खत्म नहीं हुई है, बच्चा थोड़ा और बड़ा होता है, उसकी पसंद नापसंद बनने लग गई, बट मदर कंटिन्यूज द एफर्ट। वो क्या करती है, अब सब्जी पसंद नहीं है तो उसे सैंडविच में डाल के, चपाती की तरह गोल करके, ब्रेड रोल, कोफ्ता, पकौड़ा, रायता, कुछ भी बना के नूडल्स में ही डाल दिया। क्यों? क्योंकि माँ जानती है कि अगर बच्चे ने नहीं खाया तो कमजोर हो जाएगा, तो वो अपनी कोशिश जारी रखती है। शी कंटिन्यूज विद द एफर्ट एंड मेक नेसेसरी इंटरवेंशन, मेक्स हिम ईट इन प्ले वे मेथड बाय डूइंग सम एक्टिविटीज। कुछ ना कुछ करके, चिड़िया उड़ तोता उड़ किया, कुछ एक्टिविटी लाए, कुछ गेम लाए। और मान लीजिए अगर उसके पास ट्विंस है, जुड़वा है, तो माँ का वो डबल एफर्ट हो जाता है। अब 2 बच्चे अलग चॉइस वाले, तो अलग अलग तरीके से माँ कोशिश करती है, छोड़ थोड़ा ना होती है एक बच्चे को कि नहीं खा रहे तो जाने दो। माना कि 2 बच्चे हैं तो माँ को डबल काम होगा, लेकिन आनंद भी तो अलग ही है, हैप्पीनेस भी तो अलग ही है। लेकिन अगर माँ एक स्मार्ट मम्मी है तो क्या करती है वो, शी विल कीप द फूड नियर द चिल्ड्रन एंड इंट्रोड्यूस अ लंच टाइम गेम। एक लंच टाइम गेम शुरू कर देती है, जिसमें वो कहती है कि एक दूसरे को खिलाओ, ठीक है, एक दूसरे को खिलाओ। और वह ऊपर से उनको ऑब्जर्व करेगी, मॉनिटर करेगी, ताकि बच्चा कहीं एक दूसरे को खाना गिरा न दे, आपस में लड़ न बैठे। और उनको बातों में भी लगाएगी, खाना बहुत अच्छा है, कुछ इंटरेस्टिंग स्टोरी बता देगी। इस तरह से she somehow achieves the end result that she had planned. In this case, it is feeding children. Similar is the role of teacher. एक शिक्षिका का भी यही रोल है। She must be aware of learning outcomes that has to be achieved. Learning outcome यानी जो हम किसी भी पाठ को पढ़ाने के बाद जो हम सीखते हैं, तो हम पहले ही पता होना चाहिए कि ये चीज पढ़ाने के बाद बच्चा क्या सीखेगा, उसे कहते हैं लर्निंग आउटकम। हिंदी का टर्म मुझे नहीं पता उसका। So the teacher needs to be innovative and bring in activities, playway methods, plan formative assessment, ask questions. Thus each teacher has to be engineer, यानी टीचर को इंजीनियर बनना पड़ता है। And design different ways of teaching, अलग अलग टीचिंग के और असेसमेंट के तरीके अपनाने पड़ते हैं, एक्टिविटीज अपनानी पड़ेंगी, बीच में अलग रास्ता अपनाना पड़ेगा, और make them responsible for their learning and capitalize on power of स्टूडेंट्स इन टीचिंग वन अनदर। जैसे एक माँ अपने 2 बच्चों को एक दूसरे को खिलाने में यूज करती है, ठीक है, तो एक टीचर भी अपने उन बच्चों को एक दूसरे को पढ़ाने में हेल्प करे, जो हम कहते हैं पीयर टीचिंग, पीयर असेसमेंट। आखिरी में, असेसमेंट्स आर सिंपल प्रोसीजर्स फ्रॉम ड्रॉइंग इन्फ्लुएंसेस एंड नॉट कंक्लूसिव। मतलब कि असेसमेंट जो है एक प्रोसीजर है जिससे हम कुछ रिजल्ट ले सकते हैं, लेकिन उस रिजल्ट के ऊपर हम दरोमदार नहीं रख सकते कि ये रिजल्ट इसी चीज के लिए है। हमें अलग अलग टेस्ट के लिए अलग अलग रिजल्ट डिसाइड करना होगा, और वो रिजल्ट उसी चीज के लिए होगा, वो रिजल्ट हर चीज के लिए नहीं कहलाया जाएगा। सपोज मैं एक एग्जांपल लेती हूँ कि मेरी क्लास में मुझे मैथ्स पढ़ाना है, ठीक है। अब मैंने मैथ्स का एक स्टोरी सम, जिसको लोग वर्ड प्रॉब्लम कहते हैं, पता नहीं वर्ड प्रॉब्लम क्यों कहा जाता है, मैं तो उसको स्टोरी सम कहती हूँ क्योंकि उसमें कहानी है। अब जो बच्चा मैथमेटिक्स में अच्छा है और वो अच्छा रीडर भी है, वो पढ़ भी सकता है, भाषा को समझ भी सकता है, कॉम्प्रिहेंड कर पाता है, इंटरप्रेट कर पाता है कि उसमें क्या कहा गया है, तो वो अच्छे मार्क्स लेता है। लेकिन वहीं दूसरा बच्चा जो मैथ में तो अच्छा है, लेकिन उसका कॉम्प्रिहेंशन, इंटरप्रिटेशन ठीक नहीं है, उसकी भाषा ठीक नहीं, उसको समझ ही नहीं आया कि करना क्या है, सिंबल्स तो थे नहीं स्टोरी सब में, तो उसको समझ ही नहीं आया, तो आप उस टेस्ट में जो बच्चे को मार्क्स मिलेंगे तो वो मैथ स्किल के लिए मिलेंगे, पर उसकी तो लैंग्वेज स्किल कमजोर थी, मैथ तो उसका अच्छा है। तो असेसमेंट इज नॉट कंक्लूसिव एविडेंस। हमने जो असेसमेंट लिया तो कंक्लूसिव एविडेंस नहीं है कि हाँ बच्चा ऐसा ही है, उसको यही आता है, क्योंकि हमने इसका क्या क्राइटेरिया लिया है, उससे क्या कंक्लूजन निकाला है, उसका क्या इन्फ्लुएंस ले रहे हैं हम। ठीक है, एक और एग्जांपल देती हूँ। मान लीजिए आपने बच्चों को पढ़ने के लिए दिया 100 वर्ड्स, ठीक है, ये 100 वर्ड्स लर्न करो, इसमें से मैं डिक्टेशन लूंगी। अब अगले दिन आप डिक्टेशन ले रहे हो, श्रूट लेख जो बोलते हैं, तो अब उसमें एक बच्चे ने 80, 85, 90 वर्ड्स तक लर्न कर लिए, 10 वर्ड्स उसको याद नहीं हो पाए। दूसरा बच्चा है, उसने तो 20 ही लर्न किया, उसने कोशिश ही नहीं की इतने लर्न करने की, 20 लर्न किए। अभी आपने टेस्ट लिया, टेस्ट में आपने 10 वर्ड दिए, और फॉर्चुनेटली और अनफॉर्चुनेटली जिसने 20 लर्न किए थे उसके लिए तो लकीली वही 10 आ गए शब्द टेस्ट में आपके, और जिसने 90 लर्न कर लिए लेकिन 10 छोड़े, उसके लिए अनलकीली वही 10 छूटे थे। अब रिजल्ट आया तो जिसको 90 शब्द याद हैं लेकिन उसको टेस्ट में मिले हैं 0 मार्क्स, लेकिन जिसको सिर्फ 20 ही पढ़े थे लेकिन लक या जो भी कहे उसको 10 में से 10। अब आप क्या कहेंगे, क्या कंक्लूजन निकालेंगे इस रिटन टेस्ट से? दैट इज व्हाई one test fit to all is not a good idea. There is more and more need for different types of tests, जिसे हम NEP कहता है adaptive tests, which can be changed according to the competency that we want to assess. हम जिस चीज को assess करना चाहते हैं उसके हिसाब से हम adaptive test को adapt करें, change करें, मोड़ें, उसको मोल्ड करें, और ये adaptive test student centered होते हैं. Besides, we do not blindly have to follow the NEP. NEP को हमें एकदम आंख बंद करके बस NEP है, पुस्तक है, ये गीता है, कुरान है, इसको हम फॉलो करेंगे, ये भी नहीं होगा. Each of us has to rethink different ways. अलग अलग तरीके सोचने पड़ेंगे. जिस माहौल में हम पढ़ा रहे हैं, हमारे बच्चे हैं जिनके साथ हम डील कर रहे हैं, जहां हमारा इंस्टीट्यूशन है, NEP तो एक ब्रॉड गाइडलाइन है, लेकिन NEP हमें ऑटोनॉमी देता है, हमें छूट देता है ताकि हम क्रिएटिव बने, इनोवेटिव बने, अपनी जरूरत, अपने बच्चों, अपनी वर्क प्लेस के हिसाब से. इन द एंड आई वुड लाइक टू से दैट डेविल लाइज इन द डिटेल बट अल्टीमेटली द सक्सेस लाइज इन द इम्प्लीमेंटेशन. यानी जो मुख्य कार्य था डिटेल में, वही सबसे कठिन होता है. वो तो हमने कर दिया, लेकिन सफलता तो इम्प्लीमेंटेशन में होगी. 5 साल लगे पॉलिसी मेकर्स टू इंक्लूड एवरी डिटेल इन पॉलिसी. And now it is upon teachers to actually implement these ideas. ये हमारे पर है कि हम इसको implement करें, viable बनाएं अपने हिसाब से, अपने atmosphere, अपने स्कूल, अपने बच्चों के हिसाब से. For this, the teacher needs support from their school heads, institutions, parents, community, and society at large. Institutions are as good as people you choose to run them. So the people running the institutions also need to upgrade themselves, be aware of what the policy suggests, what and how it can be implemented in their institutions, and also support the teachers. Teachers का support बहुत जरूरी है. Institutions ने अपनी teachers को support करना चाहिए, guide करना चाहिए. इसके अलावा policy says that, policy कहती है कि to understand both the students and teachers, ताकि एक बच्चे को भी समझें और टीचर को भी समझें, इसके लिए स्कूल हेड जो होता है उसने should be in touch of at least one class. उसने भी अपनी एक क्लास के साथ हमेशा टच में रहना चाहिए, उन्हें पढ़ाना चाहिए, ताकि वो ऑन ग्राउंड सिचुएशन समझ सके और सही गाइडलाइन दे सके, और जो पॉलिसी के इम्प्लीमेंटेशन जो हमें करने हैं वो रिफॉर्म्स हम सही तरीके से ला सकें. हमें एक बार फिर से सोचना होगा कि आर वी टीचिंग फॉर टेस्ट्स और फॉर लाइफ? हम सिर्फ परीक्षा के लिए पढ़ा रहे हैं क्या? खाली वही पूछेंगे जो परीक्षा में आएगा वही पढ़ाएंगे, या हम उनके जीवन के लिए पढ़ाना चाह रहे हैं? After this session also most of you will think, yeah, the policy is really good, but changing in the way teachers think does not benefit students. हम अगर टीचर्स किस तरह सोचते हैं उसमें बदलाव ला भी लें तो उससे होगा क्या? टीचर्स की सोच बदलने से क्या होगा जब तक कि we cannot change what teachers do. मतलब आपकी सोच से, हाँ मैं भी मानती हूँ पॉलिसी को, मैं भी मानती हूँ सही, लेकिन मैं क्लास में जाके क्या कर रही हूँ? क्लास में तो फिर वही कर रही हूँ. तो कोई फायदा नहीं है। आपकी सोच बदल गई, लेकिन जब तक आपने वो करना, आपके एक्शंस नहीं बदले, तब तक कोई फायदा नहीं। The national policy on education is an effort to change the way the teachers, the parents, the society think. ये सब कैसे सोचते हैं, उसको नेशनल पॉलिसी, इनकी सोच बदल रही है। लेकिन इट इज अवर रेस्पॉन्सिबिलिटी टू फोकस ऑन एक्टिंग द वे वी थिंक। ये हमारी जवाबदारी है कि जो मैं सोच रही हूँ, जो पॉलिसी बता रही हूँ, जो चीजें बोल रही हूँ अच्छी हैं, उनको मैं खाली मानू नहीं, मैं खाली बोलू नहीं कि अच्छी है, हाँ बढ़िया है बढ़िया है, इसे मैं इम्प्लीमेंट करूं, मैं अपने एक्शन में लाऊं, क्लास में जाके वो मैं करूं, तभी हमारी पॉलिसी सक्सेस होगी, वरना फिर वही दूसरी के बाद तीसरी पॉलिसी भी वहीं रह जाएगी। और अंत में श्री सुरेंद्र सिंह जी की पंक्तियां में दोहराऊंगी आखिरी में, परीक्षा और मूल्यांकन में व्यापक सुधार होना चाहिए। परीक्षा और मूल्यांकन में व्यापक सुधार होना चाहिए। बच्चे इनसे डरे नहीं, कुछ ऐसा उपचार होना चाहिए। करके सीखें, खेलकर सीखें, करके सीखें, खेलकर सीखें, और सीखें आनंददायी गतिविधियों से। मस्ती मस्ती में ही जांच परख हो, यही आधार होना चाहिए, यही आधार होना चाहिए। थैंक यू सो मच। Thank you.
Thank you, Methwani ma'am. Thank you. Thanks a lot, ma'am. It was such an interactive and informative talk. We are seeing so many compliments in the chat box, and people are really participants are really appreciating the talk, and everybody is like thankful to you, ma'am. I thank you on behalf of all of us. And very briefly, I would like to talk about the key takeaways from your session. So, ma'am, you began with the evolutionary journey of the process of evolution. That was very, very interesting and informative for all of us. And you also talked about the need for reform in evaluation system, thus examination system. It was very aptly and emphatically highlighted. Key highlights of National Education Policy 2020 were discussed. There were many points from the National Education Policy which were talked about, and also they were highlighted very aptly. Some of the points I would say that they were transfer of learning from rote learning to higher order thinking, skill-based learning should happen. And feedback of child's report card should not be judgmental in nature with regard to child's learning. Rather, it should be used for child's future growth and development, was very aptly highlighted and discussed by you. And you have also talked about that there should not be assessment of learning. It should not be judgmental in nature. It should be as learning and for learning. So the qualitative and subjective aspect, and very much, I would say, hand-holding nature of assessment with respect to child was highlighted in your talk. This was very, very, I would say, guiding for all of us. And you have also talked about that assessment should be more formative in nature, continuously assessing the child while engaging him or her in the learning. Should be I would say, philosophy of a teacher. The nature of questions that you are asking, you have very aptly asked certain questions in between, and that way the talk also became very, very interactive in nature. So the question asked by you, whether we should ask the close-ended questions and open-ended question, was also one such activity that you did in your talk. And this activity very well highlighted the fact that irrespective of the nature of questions that we are asking in the class, the sole criteria of asking any question should be whether it is promoting the thinking among the children or not. So it should not only be fact-checking in nature, which is the case maximally we see in traditional classrooms. So you try to take all of us one step beyond that and thinking about the qualitative growth of the learners. You have also talked about the diverse strategies. We should not only be dependent on asking the question for the assessment of the child. I would say that this fact was very much highlighted in your talk, that we should also think about and plan about various diverse strategies of assessing the children. They may be various tasks and various activities, not only asking the questions. You have also highlighted the relevance and significance of good classroom management, how we should take care of who is speaking, when they are speaking, and how there should be a culture of learning in the classroom. Rather than we as an external agency forcing something on child, it should be the culture of learning in class, and students should be participating on their own. And this was also highlighted in your talk, that various myths of education, we need to understand that what are the myths which are assumed to be real in nature, but they are not real in nature, and they are just myths, and we need to overcome them. And the last point, which I would say, ma'am, key takeaway for all of us was that you highlighted the fact very well, the success lies in implementation. So all the positive aspects, all the suggestions, all the motivation, what all is there in the policy can only be realized in the context when we are able to implement that. So, ma'am, on behalf of all of us, Varshney Teachers Group, and as well as the participants, I would like to thank you. Ma'am, I would just here would like to inform you that we will have question answer session towards the end of the webinar. So now we have our second esteemed speaker with us. So we'll be taking the session ahead, and toward the end of the session, we'll be having question answer. So all the participants who have question from your talk, they will be asking at the end, and I will be coordinating the entire thing. So now taking the event further, I would like to invite Dr. Sanjeev Varshney sir to please introduce Dr. Abhishek Mishra sir. Sanjeev Varshney sir, over to you, sir. Thank you, sir.
Thank you and welcome, Abhishek. We have just heard a lady from the Pune, and now it's the turn of a Pune man. So Dr. Abhishek was also in Pune when he did his PhD from the NCL, and thereafter he started his career from NIO Goa and then Goa University. So he has been to most loveliest place in India, and now of course he is working as a research fellow in Houston Methodist Hospital in the Department of Pathology and Genomic Medicine. So Abhishek is, as a teacher, of course, has been guiding MSc students and writing research papers. He's already written a number of papers in peer journals, and he's also editor of many journals, including Nature Science Report. In addition to several subject specific like Frontier in Microbiology, Basic Microbiology, Journal of Advanced Medical Pharmaceutical, Journal of Advances in Microbiology, Nutrition, Food and Safety. So he has been talking about it. But one of the passion he carries is how youngsters could be encouraged, and that is by telling popular stories. And one of such stories he is going to share with us is on CRISPR, which is how the Nobel Prize in 2020, which has gone to gene technology, that may lead to change the DNA of animal, plant, and microorganisms with precision. So Abhishek, it's my pleasure to invite you to hear and share your thought, and I'm sure you will bear a very interesting story before us. Thank you and looking forward. Abhishek.
Thank you. It's my pleasure to be here with this group. Just I keep my presentation and just a minute. I'm new to Google Meet, so I'm just trying to get.
You need to go to the rightmost button on the
Yes.
bottom side.
Yeah.
Present now.
Present now. I came and then I have to take a tab.
Select the window. Select the window.
Yeah.
And concern window where you have got your presentation already.
Okay.
Yes,
can you?
we can see it now. Thank you.
Okay. Thank you.
Right.
Thank you all for having me here. A very good evening. As you have seen a fantastic talk by Shobha ma'am, and now it's hard for me to fit in her shoes, which she has set up a very high regard. So, so here I'm to present a story about CRISPR, which won a Nobel Prize in chemistry in 2020. I chose this topic. One of the reasons is that it is a beautiful science expedition, but also this is something which is going to influence everybody, everybody on this earth, because this is the future which we are going to see maybe in 10 years or 15, 20 years, but this is something which is going to happen in medicine, which is gene editing. So in brief, most of you must know that it's a Nobel Prize, which is started by Alfred Nobel. He invented dynamite, and which made him a good fortune. And then he wrote of his will, in which he said that there would be 5 awards in the category of physiology, medicine, physics, chemistry, literature, and peace. And this will be conferred to the people who work for the greatest benefit of the mankind. Economics was added later. So one of the core concept here is that the innovation is the basic foundation for economic prosperity and better future. So let's see what these are the 2 ladies who got Nobel Prize this year, Emmanuel Carpentier and Jennifer Doudna. They developed a method for genome editing. So for those who are new to the field, I will try to generalize it. I keep my talk very generalized, and later we can take questions which may be general or maybe technical ones at the end of the talk. So when we ask who we are, what we are actually. So this on screen, you can see these are human chromosomes which are aligned here, and answer lies in these chromosomes. So we all have a pair of 22 chromosomes. Females have a pair of X chromosome and males have XY, but everything about us is coded in these chromosomes. As we know that there are environment which has an effect, and there are genes which has an effect. So the genes part comes from this chromosome and the environment. But recent researchers suggest that even environment influences these genes, and it makes imprints which may last for generations. So these chromosomes are consisting not only the genotype which is given or the genotype which we have right now, but for generations in the environment which we are living in. For example, to simplify it, like I have a hand and there is a thumb, fingers, but somebody told this thumb that you have to grow till here and not beyond this, and that's why my hand has a particular shape. It also is basically these are made up of small cells, but the cells on the back of my hand on the palm are completely different set of cells. So these all codes are actually coded in these chromosomes. Also, when we are young, when we are like a newborn baby to an old age, there are different point of time in our life. There are different expression of different hormones or different phenotypes which appear, and these are also influenced by these chromosomes, and that's what make them very interesting. So basically, if we see these chromosomes, if we can just stretch out and open it up, this will be like a 1 meter thread of 3.1 billion base pairs. These base pairs are can be abbreviated as AT and GC. A and T pairs together, G and C pairs together. So to understand the quantum of it is that you can understand that we all use computers, and computers have 2 binary units, 1 and 0, and all the coding is basically done in 1 and 0, and that's how computer system works. Now here the codes are 4 letters, so this is also called as quarantine code. Okay, So, so the kind of data, or the kind of calculation a computer can do, you can understand that if we talk about DNA as a computer, it is a very vast computer which can store a huge amount of data and which is expressed at various point of time in our life. So when we have this idea that DNA is everything, DNA is something which is influenced by our environment, our way of life, and finally what we are is DNA. So there are many mutations which causes disease, and there is always interest of people in achieving greater traits for maybe for the vegetables, maybe for human beings, or maybe the fuel which we use, those things can be improved, or the variations in the population, like we all share 99% of genome which is same, but we are different in some aspect, and these are some small mutations which may be in the genes of which are very important, or maybe in the part of DNA which doesn't code for a particular kind of protein. So to understand this, so this DNA is actually transcribed into RNA, and this RNA is transcribed into a protein which is a functional. So when we talk about our hair, skin, they are modification of a protein like keratin. We have hemoglobin in our blood, and if you see the muscles, these are actin. Even the cell, each cell of our body is made up of proteins. So the structure unit, or maybe the functional unit, maybe the units which translate something is protein, like enzymes. So this is the basic structure. So so the diseases, there are many diseases which are caused because of these mutations, and to cure this disease, there could be a simple thought that what we can do is, if we can change the DNA, we can cure the disease. And here comes the concept of genome engineering, which is like a kind of gene surgery. So there could be a wide array of diseases which could be addressed. There could be drug development studies, and there could be various kind of materials which can be made. We can improve our animal culture or the farm culture, etc. By changing these genes. But the problem is that changing these genes, changing the sequence of DNA is not a very easy process. So over the years of 20-30 years research, we know that there is a possibility, but it's very hard to manipulate these things. Some of these things which we have already seen, like for example, there are plants which are now drought resistant. There are plants which have increased productivity. There are some gene therapy for various diseases, which we know, like some of you may be aware of something called as BT cotton, which is a kind of insect resistant cotton plant, which is marketed, and improve the quality of food, maybe the size, weight, color, texture. These things are possible. So for we can take a very simple example like a mushroom. So a mushroom, which you can see on the screen, is fresh, and it becomes rotten because of an enzyme which is called as polyphenol oxidase. Now this enzyme turns mushroom, which basically is doing is aging this mushroom. So if we block this particular enzyme, what we can do is we can increase the shelf life of mushrooms. If you see in last 10-20 years, the shelf life of mushroom is actually increased in our lifetime, where we can see that that now it has been increased to 1 week. Earlier, mushrooms were not available in small towns and cities, but now we have an ability because. But what we are currently using is the inhibitors of this polyphenol oxidase. But what I'm trying to project is that if this gene is mutated in mushroom, this process can be slowed down. Now, as I showed that this DNA is a very long strand of say base pair sequences, and there would be a mutation somewhere, and it is very difficult to find out where this mutation is. Then it is very difficult to find and correct that mutation. It's like you have a very big fat book, and you have to find a particular word in that book, and it becomes very difficult to even find it out. And so over the years, there are technologies which are developed. For example, exactly 20 years back, we got Human Genome Project, which was something which fascinated people of my generation, taking up biology as a subject of research or study, because at that point of time, people were all the newspaper of like 2000 were filled with something like the ease. Now I can talk about DNA is actually like back there when people started talking about DNA, chromosomes, so much, because 20 years back when Human Genome Project come, everybody was excited that we know what are the exact base pair of the genome. But what we don't know is, and people were very enthusiastic about it, that if we know the base pairs, we will know what disease, what gene causes what disease, and we can cure them. Later, people realized that this is much more complicated, and then they started dwelling it. But still, we don't have something in which we can do it in vivo. In vivo means in like a live organism. We cannot easily change this DNA. There are already some techniques were there, which is like this zinc finger and tail. So I will not go into the technical detail, but basic idea is that these are protein based. So if you want to modify some strand of DNA, there should be, you should know which page you have to go, and these machines will help to reach that spot. But because these are protein, it is very difficult to design a protein which will identify a particular sequence. There are some proteins which are there which can identify particular sequence, like these zinc fingers and talons. But the problem is that I want a particular letter, for example, say any word like protein. So I want to search in a book word protein. So if I have a zinc finger for it, it is easy for me to just find the word protein. But again, now I have to check another word, say genome. Now the same machine cannot be used, and now because naturally existing machines are mostly protein recognizing, it is difficult to identify the word genome. So I have to change this and design, and designing this is even very tough, because protein is something which is complex structure, and to achieve this particular complex structure is very difficult. Now this CRISPR, why it is very important now is that it is an RNA base. So RNA is, as you remember, is something comes from the DNA. So the sequences are very similar. So an RNA can identify DNA just by pairing. If you can see the diagram, this red color thing can pair with the blue color thing. The reason is that they have similar kind of ATGC sequences. In RNA, there is no T, there is U, but they identify A, G, C, U, and T. So, so in this way, what is possible now is easy to find in a book. It's to simplify this, you can understand that you need to find a word in a big fat book. You have to go through every page, and it is relatively difficult in comparison to the present technology, which is RNA based, in which you can just do a control find, control F on a computer, and what you can get is your exact word. So this technology is basically a long acronym of CRISPR, is clustered, regular, interspace, short, palindromic, repeat. I know it's complex, but you don't have to understand what it, you don't have to remember the whole acronym, but understand what it is about. So basically, this CRISPR thing is derived from bacterial immunity. So like viruses, which can attack our body, there are viruses which can attack to bacteria, bacterial cell too. And as you know that we have a defense system of our which can kill this virus, which can restrict. Nowadays, people are much aware about virus because for coronavirus, we know that there are 70–80% people who can control this virus, but there is some population which cannot. But what I want to emphasize is that there is a system in our body which can restrict virus, and same kind of system is there in bacteria, which is called as CRISPR. So how it is seen is that so there is a RNA which has if you can see here Cas9 in pink, yellow. So this array is of the protein. So when this translates, this will form a protein, and it has something like repeats. So you can see blue, green colors on the right side. These are the RNA sequences of the viruses. So whenever a bacteria is infected by a virus, this new color formation is it tells bacteria that it has been already infected by different sequence, so that it just keep this sequence into the array. And whenever new virus comes, it identifies if it matches, then what it will do is that it will transcribe this, and you can see this Cas, which I said is a protein. Now this protein will come out expressed, and it will identify these RNA. So you can see that there are different kind of RNAs. Array is here, which is marked in green, yellow, red, pink. This will be distributed, and now if in the genome, if there is same virus which attacks it, so what it will do is it will go and recognize. How it will recognize? Because the sequences are same, because it is derived from the virus, and this protein will act like a scissor and it will cut down that virus DNA. So this was the basic science or basic existing system. Now what is possible is what scientists have done is this Cas9, this big purple block, is a protein, and this red color thing is a DNA. This blue thing is something which can identify this DNA sequence, and then if it identifies, it makes a cut. Okay, now if it makes a cut, if we provide a donor DNA which looks fluorescent, what we can do is we can introduce a new gene sequence into the system, and this can be expressed in human or in cells or in the animal system. And you can understand, like for example, there is some gene which we know that this gene will cause cancer. So what we can do is we can disrupt this gene, or we can rectify this by changing the sequence. So one thing is that if this gene is there which may cause cancer, we can disrupt it and put something so that this doesn't work, or we can repair this sequence or change this sequence, like is shown in the diagram, that these sequences are changed, and then what we can get is the repaired function. So overall, so overall, what I want to emphasize is this is possible. And now what we can do is we can do it in the organism which is already grown up, like mice, or in some cells, or into the cells of like germline cells and all, which I'll come in a minute. Now the story is about this timeline which starts. So if you see that the idea of CRISPR, what is this CRISPR? It comes in 1987. So the CRISPR is, as I told you about the acronym, is basically some repeated DNA. So some scientists find out in 1987 that there are bacterial DNA which has these repeats. So what? Yeah. So what? So there are repeats, and nobody can think of that it has. So there are DNA and there are repeats, which is something usual. And you can see that to next 13, 14 years, nobody even bothered about it. And later on, people started understanding what these genes are about. So what I want to emphasize here is that when some research, some basic research starts, we don't know that where it can lead. So sometime some research are labeled as why we need to do this, why this kind of knowledge is important. Well, we don't know right now why this is important, why we need to, why these questions are addressed, but there may be a good outcome of those, or it may be like a foundation block for a research or a translational technology which is going to happen years later. It's like Newton's law that apple is falling on the ground. So what? So everything falls on ground. What is the significance of it? But now today we can do this conference because of some satellite which is in the space projected using this concept. So here what I want to emphasize is that a very simple thing, that analyzing gene sequences is something which has led to the foundation of this technology. And even later in 2005, people identified that these spacer sequences are for immunity of bacteria against some kind of viruses. And there are a significant amount of research which took place, like how this enzyme works, what is the structure of this enzyme, and so on. And later, the Nobel Prize, which is given to the two ladies, Carpentier and Doudna, is this research, which is highlighted here, is 210 and 212. And so this story is, when we talk about the story about Nobel Prize, it may be because of one or two incidents, but these incidents are actually programmed a lot earlier in the system by peer scientific research. And later on, when this thing that the ability of the system to work and cleave and repair is done in 2014, there are at least 20 papers in different journals which appears and which tells us about that it can modify the plants, animal cells, and so on. So this technology is mostly the contribution of these three people in this. Jennifer Doudna and Carpentier, you know that they received a Nobel Prize, but also there is a guy named Zhang who first time showed that this bacterial system can be effectively work in eukaryotic system, which means the animal system and the plant system, plant cells, and this could work. And what we can do is the genome editing. So there was a long patent war between these two who belong to University of California, Berkeley, and Zhang, who belongs to Broad Institute. Later on, the patent war was won by Jennifer Doudna and Carpentier, and so the Nobel Prize. But the importance of the research of Zhang cannot be neglected. So if you have any questions particularly, then we can take it at the end. But I want to give a general overview what this is about. But my main aim is to project this, that where we are heading, that what could be the healthier and prosperous future. So as I said, that in my talk, that this is going to influence everybody. It doesn't matter whether you are a science enthusiast or not, but medicine is going to change step on step. Like, for example, this is some of the data which we can see on the screen. So there are different set of diseases which is like cancer. So there are 15% of cancers in which the gene therapy clinical trials are going on. There are a lot more diseases which are genetic diseases, skin diseases, metabolic disorders like diabetes, blood pressure, and all these studies are in clinical trials for this. So the future which we are expecting is that there are many types of cancers which could be cured, and this will not happen in like 50 years or 60 years down the line, but something like which is going to happen in a short span of like 10 years. If you can remember the website, the internet, that in 90s not many people are connected, but in 2010 everybody is connected. Like the mobile phones, in 90s very few people have mobile phone, and one of my teachers said that you all in 2002 that you all are going to get something called as mobile phone. At that time we don't have a understanding what, I have not seen even a mobile phone in 2000. But she said that you all are going to get this in say 1 or 2 years, and nobody believed it. 4 years later, everybody in the class had mobile phones, and that's what is going to happen now with the medicine. That now we have sequenced the human genome 20 years back. We'll come to it again. So what achievements have already been set? So using this CRISPR genomic editing, some diseases in animals are already cured, like liver diseases, which are by gene editing and not by using drugs. And this set of system is people are trying to get, because we cannot do research on human. So these things are still in clinical trials, but you can understand that 2012, 2013, when this technology is just discovered and people understand how this gene editing is possible, and we can see from that aspect that it is just 8 years and we have so much stuff already in the market. And there are many companies, pharma companies, which are using this technology to be developed for various diseases. Another important aspect is organs. So we know that there is, though there are donors, but there is always shortage of organs like liver, kidney, and so the consequence of any metabolic disorder, like say diabetes. So diabetes itself is not a killer. Diabetes itself is not a big problem if it doesn't start hurting your tissues or organs like kidney and liver. So most of people who suffer from diabetes are not affected because of the level of sugar, but the consequence it leads. So so the organs. So what this gene editing can do is it can generate humanized pigs. Humanized pigs means the pigs which will have an organ, like for example liver, and it will be a human liver in a pig. So what we can do is we can cultivate human organs in animals, and this is possible because of this gene editing technology. Also, the biggest breakthrough which came in this is that this system works in human. So in 2015, one set of Chinese research group, what they used is something called as non-viable human embryos. So we cannot do research on human embryos for ethical concern. So these non-viable human embryos are basically something which are leftover of IVF technology, which in vitro fertilization. So the leftover of this, some of these embryos are taken up, and they are studied, and what they found is that they can modify embryos, and this is being a big news, that what we can do is we can modify human embryos or human cells to that extent, and that's what we are understanding it as future. Now there are groups in UK which are doing same thing, and this kind of research is in progress. Now what I was talking about human genome is this. So if you see 2001 on the extreme left, this is the year when human genome was out, and you can see the cost of this genome. It is $100 million. Means if you have to sequence one human genome, it will cost $100 million, which is very expensive. It's not possible for somebody to afford, unless I don't think anybody would do, except like people like Ambani's or the big giants can afford it, but it was not possible in 2001. Now, if you see, the cost of this is down to $1,000, which if I translate, it is just 70,000 rupees. Now, I know that 70,000 is a big amount still, but this is a process. This is in 2019, but the same story will not be there in 2022 and 23. So you can see there will be a drop, and it will come down to $100. Even there are some system which not in clinic but in laboratory system which can generate human genomes at a cheap cost like $400–500. Now this line you can see the Moore's law, which tells us about. I'm not an economist, but what I understand from this law is that everything is going to get cheaper when we progress in technology innovation. So for example, the cost of television, say in 90s, and the television which you have right now has almost same cost with much advanced features, and so at particular time the cost will keep on decreasing. But you see the decrease in cost in genome service from $100 billion to $10,000 in till 2012, and which is a steep fall. And because of this steep fall, what we can predict is that human genomes will be easy. So it would be something like you go to the doctor for an annual checkup, and what you're going to do is other than your blood test, you're also going to get your genome, and you do it once, it is with you for lifetime. And what would be the cost? Cost would be $100, like say 10,000 rupees, which would be a reasonable cost in 2025. So what I'm trying to push is there is a great opportunity which is coming for everybody, for whether you have a business and you are interested in some sort of innovation. So these are the area where which you can look into, or you are just a general individual who want to get your genome done. And there are many companies in India, which I'm not putting it here right now, but there are many companies which are providing genome services. And so as of now, their cost is like $1,000, which is like 70,000-80,000, but it is surely going to cut down to 10,000. And these technologies are going to lead medicine in future, which I want to show it with this diagram of the mobile phones, which were in 90s, and this is the one which we have right now. And we know that, and we have seen this evolution in half of our lifetime. Like if somebody who born in 90s would also have seen this substantial growth in mobile phone, internet. And now this would be the time for medicine, and same kind of evolution we can see, evolution, or precise term would be revolution of medicine, which is possible. So I will end here my talk, and I will happy to take questions and comments on this.
मजा आ गया भाई.
Thank you.
Yeah, really, we'll have a longer question answer session after this talk. Yes.
Abhishek, you may stop sharing the screen.
Okay. Yeah.
Thank you, Abhishek, sir. I would like to give you thank you for this informative lecture and talk. So, sir, we really thank you for this enriching and informative talk. We would also like to thank you for coming down at the level of audiences, I would rather say, sir, because this topic was not so easy for everybody to understand, because everybody may not be specialized in the area. But I really appreciate on behalf of all the participants that you have tried your best to bring down to the level of audiences and make it as simple as possible. And I think most of us were able to understand the talk. So this is something which is very good for all of us. Some of the key takeaways from your lecture were history of Nobel Prize, sharing of the pertinent information of Nobel Prize 2020 in the area of genome editing, concept of genome editing, genome engineering, human genome project in the year 2000, simple discussion of various concepts such as nitrogen base, RNA and protein, CRISPR bacterial immunity procedure and related research data over the years. And at the end, we could also see the optimistic contributions of gene editing biotechnologies in the near future. So this is something which is very, very informative for us, and the lecture has been very interactive, sir. So once again, I would like to thank you on behalf of everybody for this enlightening talk. With the permission of senior members, I would like to proceed now for the question answer sessions. Sir, thank you, sir. Thank you, SK Varshney, sir, Gagan, sir. So we have many questions in the chat box. I would like to invite both the speakers to be with us so that we can ask questions on behalf of participants. Just give me a second. I would read the questions from the chat box one by one. So there was one question in the starting for Shobha ma'am. Shobha ma'am, this question was for you. From Umesh Chandra Saxena ji, he is asking, would teachers need to be specially trained to implement adaptive assessment? Ma'am, I would repeat the question. Would teachers need to be specially trained to implement adaptive assessment?
The word adaptive itself means that the teacher has to adapt the assessment mechanism according to the situation she is in, according to the children she is teaching. So it has to come from within the teacher, from her experience and from her interaction with the student. But मोटे-मोटे तौर पे टीचर को ट्रेन किया जा सकता है असेसमेंट मैकेनिज्म पे, ठीक है ना, कि असेसमेंट कैसे लेना, वो इस तरह से वर्कशॉप्स में, टीचर ट्रेनिंग्स में, बीएड कॉलेजेस की एजुकेशन में वो सिखाया जाएगा. लेकिन वो एक तरह की असेसमेंट्स हो गई. लेकिन हम बोलते हैं उसको अडेप्टिव इसलिए है कि उसको चेंज करके हमको अडेप्ट करना है, चेंज करना है अपने माहौल में, अपने स्कूल में, अपने एरिया के, अपने बच्चों के साथ. तो अडेप्टिव को स्पेसिफिक ही अडेप्टिव है करके नहीं सिखाया जाएगा, वो टीचर ने अपने इंटरेक्शन से, अपने अनुभव से उसको अडेप्ट करके चेंज करना होगा, जो बेसिकली असेसमेंट मैकेनिज्म होंगे। ये मेरा।
Thank you, ma'am. Thank you. I hope the question has been answered well. Now, moving on to the next question, ma'am. This question is from S.K. Varshney sir, and he is asking, Shobha ji, you talked about talented children, but what about children gifted with a virtue? There may be passionate about singing, music, performing art, designer. How they could be nurtured, assessed, and promoted in school culture?
हाँ, यही तो नेशनल एजुकेशन पॉलिसी 2020 खास लेके आई है। यही तो चेंज हम खास करके कर रहे हैं कि नाउ देयर आर नो साइलोज, कोई अलग कंपार्टमेंटलाइज नहीं है स्ट्रीम्स में, यानी कि आपको म्यूजिक, आर्ट, डांस ये सब भी अलाउ होगा, आप किसी भी स्ट्रीम के साथ कर सकते हो। फिजिक्स, केमिस्ट्री सब्जेक्ट के साथ आप म्यूजिक भी ले पाएंगे, आप किसी कॉमर्स के सब्जेक्ट के साथ आप डांस, क्लासिकल डांस ये सब भी रख पाएंगे। इसकी अनुमति, ये साइलोस ब्रेक करने की फैसिलिटी पॉलिसी ने दी है। अब ये हमारे पर है, हम स्कूलों पर है, इंस्टीट्यूशंस पे है कि वो अपने स्कूल में वो अवेलेबिलिटी दें बच्चों को। तो जैसे मैंने कहा ना, सबको मिलके चेंज लाने होंगे, तो स्कूलों ने भी अपने स्कूल में पॉलिसी तो बन गई, कानून तो बन गया, अलाउ किया, परमिशन दे दी, लेकिन प्रोवाइड तो अल्टीमेटली स्कूल ने करना पड़ेगा। तो वो इन्होंने स्कूल में टीचर्स प्रोवाइड करने पड़ेंगे, और हर स्कूल के लिए हर टीचर रखना पॉसिबल नहीं है, तो उस एरिया में ऐसा कुछ हमें अरेंजमेंट करना पड़ेगा कि ये स्कूल में ये वाले सब्जेक्ट्स अवेलेबल होंगे, उस स्कूल में ये वाले सब्जेक्ट्स अवेलेबल होंगे, और उस हिसाब से बच्चे वहां जाएंगे जिन्हें वो शौक होगा। लेकिन पहले जिस तरह से अलाउ ही नहीं होता था कि कॉमर्स वाला बच्चा ये ले सके, उनको उसी 5 सब्जेक्ट में से 7 सब्जेक्ट में सिलेक्ट करना होता, वो अब पॉलिसी अलाउ कर रही है। तो स्लोली एंड ग्रेजुअली डेवलपमेंट होंगे और ये सब सुविधाएं आ जाएंगी, अगली जनरेशन के बच्चों के लिए बहुत अच्छा होगा, ऐसा मेरा मानना है।
I think S.K. Varshney sir wants to say something. Sir, please go ahead, sir.
नहीं, मेरा क्वेश्चन दूसरा था मैडम। मेरा क्वेश्चन ये था कि जैसे कि कोई बच्चा है जिसके किसी भी चीज के लिए पैशनेट है, बट जो एकेडमिक सब्जेक्ट्स हैं उसमें वो उतना अच्छा नहीं कर पा रहा है।
हाँ।
तो जैसे दुनिया में कई जगह हमने देखा है ऐसे स्कूल्स होते हैं कि अगर मान लीजिए बच्चा फुटबॉल के लिए पैशनेट है तो उसको फिजिक्स, केमिस्ट्री, मैथमेटिक्स, हिस्ट्री की कोई जरूरत नहीं है, एंड ही कैन परस्यू हिज पैशन। बट हमारे यहाँ वो स्कूल कल्चर में वो चीजें नहीं है। तो अभी हालांकि ये थोड़ा सा गुंजाइश आ गई है कि along with academics he can pursue the passion, but एक माहौल हमें उस तरह का भी चाहिए होगा कि अगर पैशन है जिसको निखारा जा सकता है, पॉलिश किया जा सकता है, और एट द कॉस्ट ऑफ एकेडमिक्स। अभी एट द कॉस्ट ऑफ एकेडमिक्स हमारे कंसेप्ट में अभी भी है।
अभी तक तो हमारी सोसाइटी में इतना एक्सेप्टेंस नहीं है, एट द कॉस्ट ऑफ एकेडमिक्स हम नहीं अलाउ करते हैं, है ना। पेरेंट्स आज की स्थिति में, मान लो मैं जब स्टूडेंट थी तो मेरे पेरेंट्स मुझे कभी नहीं अलाउ करते कि मैं पढ़ाई छोड़ के सिर्फ संगीत ही लूं, लेकिन द थिंग्स आर चेंजिंग। अभी मेरे ही जानने वालों में एक है जिनके वाइफ डॉक्टर है और हस्बैंड एक बहुत ही ऊंची पद पे एयरफोर्स ऑफिसर है। उनका बेटा, उन्होंने सिर्फ संगीत करता है, म्यूजिक, क्लासिकल सीखा है, इंग्लिश भी गाता है, और यही क्योंकि वो पैशन लेके चला, लेकिन फिर भी 12वीं या आई थिंक ग्रेजुएशन भी नहीं किया, उसने 12वीं तक पढ़ाया है, और अब वो म्यूजिक ये अपना पैशन लेके उसको ही अपने प्रोफेशन लेके बनाएगा। तो ऐसे अभी चेंजेस आ रहे हैं, थोड़ा समय लगेगा, क्योंकि अभी तक हमारे देश में म्यूजिक या स्पोर्ट्स या इस तरह से लेके जब हम आर्ट लेके चलते हैं तो उतना अर्न नहीं कर पाते हैं लोग, और हर जगह पैसा तो चाहिए ही होता है। दूसरे प्रोफेशन में जो टेक्निकल पढ़ाई से जब जाते हैं तो अच्छी इनकम हो जाती है, और पोस्ट जब स्पोर्ट्स छूट गया उसके बाद का भी फ्यूचर के बारे में अब तक पेरेंट्स डरते थे, लेकिन अब द चेंजेस आर कमिंग, पेरेंट्स में एक्सेप्टेंस आ रहा है, पॉलिसी में चेंजेस हो रहे हैं। तो ये जो आप चाह रहे हैं कि बता रहे हैं कि ये ऐसा क्या होगा, तो ये बहुत जल्दी होगा, ऐसे आई एम वेरी ऑप्टिमिस्टिक अबाउट इट।
Thank you, ma'am. Thank you for answering the question. I think Gagan Gupta sir also wants to say something.
No, I mean, sir, I mean, just to add what Shobhaji is saying, and probably to precisely to the precise answer of your question is, yes, now this higher education has is expected to give some situations where suppose a child is doing extremely good in one subject or in one stream, then one can afford not even good marks in other subjects. At this moment, the benchmark is 33%. So though these 33% will remain in principle, but if somebody is doing say 80% in one subject of interest and say 15% in some other subject, so that can be allowed. So this kind of number one. Number two, as far as the ideas like KVPY, NTSE, Olympiads, etc. are being given in this policy at school level, unfortunately, at this moment, we are not seeing for all kind of courses. Coming back to your question, nurturing the gifted children. I hope and I am very optimistic that in days to come, when we are going to talk about how this policy will be implemented or transformed into the school system, then we will probably see that these kind of things will come on. I have, yes, I had this question in my mind and I have written it in my notes. So as and when I work in, Abhishek ji, I work in NCERT here. This is the organization which is primarily responsible for all these kind of designs. So yes, these questions are there, sir. Probably I have been able to answer some of your query to some part.
And we can discuss it much later.
Yes,
Move forward.
Yes.
Move forward.
Yes, sir.
So a very interesting discussion has been started and we are looking forward to take this discussion further. Moving ahead, in the question and answer session, there is a question from the name, it is showing Mr. AK. So the question is for Dr. Abhishek Mishra sir, do genes play any role in passing on mental health issues like depression from one generation to another? Okay, Gitanjali, below it's written Gitanjali. So Gitanjali ma'am is asking, do genes play any role in passing on mental health issues such as depression from one generation to another? Abhishek Mishra sir.
So I'm not an actual expert in understanding psychology, but genes play the role in multiple ways. Like for example, that there could be some psychological issues which are because of some genes associated with it. Now there is something which is called as epigenetics, which means there are some modification to DNA, which is basically some methyl group which is added to this DNA, and this happens for most of genes. So some genes can be silenced and some can be activated. And what happens that these kind of addiction can go to generations. So not what I'm trying to say, it's not only DNA, but if there is something imprinted in DNA, may also go for subsequent generation. So so for example, like in psychology, it could be like a brain development function, or it may be because of some hormonal imbalance, or there could be different reasons for it, or maybe some kind of drug usage. So these things generally imprint and it goes for next generation. So yes, there are many there are effects of gene.
Thank you, sir. There is another question from SK Varshney sir. The question is genomic engineering and ethics. How do you see it? In India, genetically modified crops was socially rejected. What is the current practice in US? How do you see future of personalized medicines?
These are some interesting things which I want to address actually. So one of this is the genomic, and it varies from country to country. And there are laws and there are discussion, ethical committees, and people are coming to one thing for sure, is that this decision is left to the consumer. Okay, so finally what is going to happen, yes, there would be, but what progress which we have seen in maybe in India or in US is that you mark them and mark with authenticity. So there should be, you can mark that these are gene modified vegetables and these are not. So it is up to consumer which one they choose to use, and they should have the knowledge that what they are using, instead of just flooding vegetable markets with some tomato which is gene is modified, and some people who are not interested in that, or it's can do it. Now there is another thing is, most of these modifications which genomic modification which Sir is talking about is taken by a viral vector. So what they use is they use a viral vector, in that they insert DNA and then they infect the virus to the plant cells, and this virus DNA is integrated into plant genome. So this is one of the reason that people are more conscious about it, that what if this virus spread is there, or if this modification goes wild, like you modify some plants and it goes into wild habitat, and it may affect the other natural vegetables, which should not happen. So the containment of this, so if there is a defined policy that these vegetables can be contained in a particular region. And the second thing is, which is most important in this particular thing, is that what is good. Like you can say that a tomato which appears red, bright and big is good, but that doesn't mean it is nutritious also. So we are picking and choosing the attributes, and that has to be very conscious decision. And that's why genetically modified plants, we have not seen it, though we have it for like 20 years, but we don't have a big market of those plants. But there are 2-3 things which are going to change. One is use of viral vector. So with CRISPR, we know that the delivery system may not be as typically as virus, and it would be an easy delivery system. So the problem is that in plants it is difficult to deliver the genes. But if we can have this, and it can be modified, which can be lost after some time, there are things that it can be done. So there are progress, there is consistent thing, but the ethical issues is huge. Like for example, like similarly for the medicine, like we know that we can clone humans. Though we don't have an experiment evidence, but in 90s there was a sheep which was cloned, and so same kind of things can be done to human. We know that IVF is there, and they use a similar technology, so it is possible. But there is an ethical issue. You cannot grow a human and take its organ when it is needed, because that human has life now and has some rights in each and every country. So the possibility of doing that, but we are reaching somewhere in between. जो बीच का जो इसका रास्ता होगा वो ये होगा कि can we culture those kind of organs in the experiment animals. So these are not wild type pigs, these are lab pigs. We can generate these kind of modification in plant for slight improvements, like for some modifications which are reasonable and not completely changing the plant, and go step by step with very strong policies and the perspective from the society as well as scientific community. I hope I've answered most of it.
Thank you. I was also looking for personalized medicine.
Yes, that is very interesting. So personalized medicine, if you see that, like if we see 20 years back, there were very less people who were just visiting doctor for no reason, like for annual health checkup. So 20 years back, or until unless you are very old, nobody is going to visit doctor. But now, what we have seen that there are more number of tests, and what we are trying to do is by these tests is early identification of disease, right? So if somebody who is like 35-40 years and borderline diabetes, and what they can do is they can improve their lifestyle, uh, and they can maybe prolong the time when they can be actually affected. So same way, if we have genomics in place, then these things would be more more effectively done. For example, now also personalized medicine is there for cancer treatment. So like there are several cancer treatment, there are various drugs. There are many drugs which don't work for all the cancer. There are many treatments, like for example, there is a specific lung cancer, but there are 3-4 drugs, and some may work, some may not work. So for cancer treatment, for acute diseases, we are in personalized medicine. So if these things, because these are costly things, so if it requires cancer cell culture treatment with various drugs, and see that which one works well, and then use it that one. So this is an expensive thing which is not possible at present. But what I'm expecting is in next 10 years, it would be very much possible that people are going for personalized medicine. Or for example, the dietitians, nutritionists, their role are improving on the basis of, again, it is personal, or maybe a gym trainer or an athlete. So they have a specific kind of nutrition program and all. Now, if we can integrate this with genomics, it would be that they can make more rational decision. They can make an early prediction. So yeah, I think predictive medicine or precision medicine is going to be future, and it would be like for everyone. Like it's not for elite. It would be like for general population. In 10 years, we will reach to a point where we are into precision and predictive medicine. Personalized the system which we are looking. Yeah.
Thank you, sir. There are 2-3 observations come question by Gagan Gupta, sir. So he is saying, I expect the treatments may become cheaper, that you have already addressed. And he has also observed that I hope that this project would not have any ethical issues associated, and these are with gene cloning. And he has also written $100 million to $1,000 in just 19 years. What about the time involved in identifying the genome? Maybe few minutes. So
yeah, that's a very, very good question. Because I didn't address this, but this is also an important thing. So Human Genome Project started in 1990. And the first genome which came out is in 2000, which means it took 10 years to do a genome sequencing. And at present, it will take 24 hours. 24 hours. So if I get sample today, in 2 days, we'll have complete sequence achieved. So this time is also a very important factor, which we can say that we have taken a long leap in these 20 years. Yes. Yes, Gagan, sir.
Yes, I have. See, you in between in answering the question, Varshney Saab's question, you talked about predicting the health of a person, or I mean, at an early age. I remember Benjamin, who got Nobel Prize somewhere in 2007, Israeli in chemistry, and making a statement that probably through this gene identification, there is a big question: are we going to improve the life, or we are going to disturb his life by telling him early that this is what is going to happen to you at the age of 70 or at the age of 50 or at the age of 40. So this is one issue. I am number one. Number two, is there any technological limit in identifying the zone? You said that 24 hours. I mean, with the advent of new technology, maybe is there some possibility of getting it in 7 minutes or so? Because I remember having read somewhere, I do not remember exactly the reference today. Once it was being said that up to 70 minutes is probably the limit below which this identification is difficult to take place. So has there been any change in these numbers?
So, for the timing, so there could be different kind of sequencing. One could be few gene based, so it can take in minute, but the whole genome, the whole base pair identification, it takes a longer time. They have to make different. So this is, if we take out this DNA and stretch it, it would be like 1 meter of thread. So it's a very long thread, and what they do is they chop this into smaller pieces, and then they do sequencing for each fragment, and then they compile it together with computer databases. The second thing is, even if you get it in, so the second limitation is, so even if you get the sequence, we don't know, because we just have 80 GC thread, a thread of 1 meter, but the interpretation, the analysis, it takes longer time. Now, second, now this is also important thing, that that whether it would be intervening. So mostly what medicine is focusing is on the diseases which are very severe diseases, which are not easily cured. So if like right now also, we go for a blood test, and if somebody is young, say for example 30, and his blood pressure or sugar is high, doctors will not put him up on medicine, but they will say that you can improve, you have some time. So that is also an important aspect of health, that you don't have to modify all the time, but if you get an early indication, what you can do is you can improve. And finally, what we have to is take some middle path, which is using this knowledge wisely, with with all the ethical and social issues addressed.
Thank you, Abhishek sir. I think there is last question for you by Nishant Varshney Sarji. He is asking, do you think type 2 diabetes can be treated with CRISPR technology?
There are already published setup in which they have shown this in mice. So when we say this type 2 diabetes, there could be two things. One is the patients are not able to produce enough insulin, or there are receptors which are there to receive insulin. They are got damaged over the period of time, or they are not able to sense insulin. So what CRISPR can do is, which I have not shown in my presentation, is it can do some kind of gene upregulation. So it can do some modification in which some genes can be upregulated or inactivated or modified. So modified for the receptor, and for insulin production also, what they can do is they can modify it for more insulin production within the human. But then again, the thing is that if there is a smaller problem, you don't use a big machine, right? So there are methods in which there are humanized insulin is produced. So if you if you talk to the older people, you'll find that in olden days, when somebody is suffering from diabetes, they have to take very regular injections, and still the disease severity cannot be avoided because there is some sort of insulin resistance, which appear because of regular consumption, and those insulin was not as good as present insulin, which is absorbed by the cell more quickly, more often. Even the drug which is metformin right now for diabetes is an improved version, and it has better absorption and effectiveness. So those people like who I'm sure many in this talk will be there who can see that there were people in 70s when in 1970s and around, when there is diabetes, they have to there is some problem with their digits, finger digits, they get roded, and more of cardiovascular problem very early coming, which has now been improved a lot. So so with CRISPR we can produce more humanized insulin. So we don't have to be taking some insulin which is non-human or cloned in E. coli. We can have something which is very close to human insulin. Yeah.
Thank you, sir. Thanks a lot for answering the question. I think next two questions, these are the last two questions also, are for Shobha ma'am. So Shobha ma'am, I'll be reading the questions which have been asked for you.
Priyanka ji, Shobha ji has left.
Okay, okay.
But
So
we can still answer this question.
Okay.
Please read that.
Okay, okay, sir. So, sir, there are two questions for Shobha ma'am. One question is saying that by Supriya Shinde ma'am, what measures are taken to upskill the teachers? This is one question. And Nishant Varshney Sarji asking, do you think our professional courses will have the same flexibility with their curriculum like NEP? I mean, today engineering colleges are asking PCM for getting admission, or medical colleges need students to have been studying PCB. What if students have not taken any of these required subjects in their school?
So, on behalf of Shobha ji, may I respond to it? Nishant ji? Thank you. The first is about the teachers upskilling. So, this National Education Policy says about in-service training programs and pre-service training programs both. For in-service training programs, every year 50 hours of professional development training programs are mandatory for every teacher, are going to be mandatory for those teachers. CTET, or the common test, common eligibility test for the teachers, is going to become compulsory for all. And there are different measures. B.Ed programs are going to be embedded or clubbed with the content, with the 4-year BSc B.Ed, BA B.Ed, BCom B.Ed programs, etc. So this is going to upscale the teacher's quality in nutshell. It is expected. Number two, yes, Nishant ji, that there are already conversations going on, but some time will take. I mean, give some time. AICTE has already started discussing about leaving chemistry for the moment for the test, for the engineering colleges entrance examinations. These medical colleges are yet not responding to this. They say that the kind of physics, the kind of chemistry, the kind of biology which is required for them is still necessary. In fact, they are asking, I am a physics student, so they are asking us, they are putting us hard that increase the syllabi of physics, because they do not have any physics faculty with them. So they do not have any mechanism in upgrading the children, the incumbents, as far as physics is concerned. So the flexibility, yes, I do not see much hopes about this, but some flexibility will come. Obviously, some course will always have a prerequisite. Today, the child who is doing music, for example, is not will not be able to shift to the medicine tomorrow without having done some certain amount of prerequisites. I'm not much hopeful about it, but some flexibility will come. The NTA and the National Testing Agency is, we are discussing it with the NTA, but at this moment, I cannot tell more. Nothing concrete has come out. Nishanji, I am not able to answer you fully, but
no, it's fine. I think fine, because this, as far as this medical is concerned, I think this chemistry and physics are both are important as well, like biology, because this biochemistry and biophysics you have to study. So human cells is human body is more like biochemistry. Actually, everything is like disease and biochemistry. So I don't think so. Leaving a chemistry will be a good idea.
No,
like,
they are doing it for AICT, for engineering colleges.
Engineering college,
they are considering it.
Maybe they can get away with the chemistry without having a chemistry. But the problem will be like, because like if we have the person, like student has made up a mind, like he want to do a medical, so he has to take those PCM or PCB in the 11th or 12th. The problem is that like once like it will be like easy to switch over.
That will come. That
has like say he has taken he has a music. Okay. Like, he has taken a music in 11th and 12th, not a chemistry of biology. But then later he wants like I want to do medical. Say I've developed an interest in medical now, but he has not studied a chemistry or biology in 11th and 12th. So that like he has to study those subjects again for couple of years.
This cannot be. Some certification would still be required, but flexibility will come 2035.
Okay.
2035. I mean, it is difficult to see all these kind of changes in practice before 2035.
Because I remember, because my wife is a teacher now. She's studying 5th and 6th in Bangalore school, and her schools has already asked. I think maybe they may implement this NEP from next session. So I'm not sure whether the practicability will means like whether the implementation will be far away or still they're doing it.
No, I mean for this particular kind of thing. 2035 is the limit, but I do not expect anything to come out constructively or concretely before 2030.
You mean for this engineering or professional colleges? Okay.
For this medical colleges kind of thing, you said that the child, yes, prerequisites are required. Medical colleges cannot teach physics in their curriculum. Cannot. They are yet not ready.
Sure.
They are yet not ready.
Thank you, Gupta sir. I think there are no more questions. All the questions.
If anybody wants to ask,
if somebody wants to ask question, they can speak. They can say it.
Raise the hand.
Yeah, they can raise the hands also. Yeah. I think, sir, there is no question. So, sir, please guide. Should we move towards the vote of thanks?
Yes. Abhishek ji, आज आपने हमारा Saturday बना दिया और शोभा जी तो अभी नहीं है. This is on my personal behalf. Yes, I learned a lot.
Thank you. Here, sir, same here.
Thank you for having me here.
Now we would like to have you again.
जी sir.
Yes, सविता जी से request करते हैं.
जी sir. So, taking the event further, I would like to request Savita Varshney ma'am to please come on the stage and propose a formal vote of thanks. Savita ma'am, over to you.
Good evening, everyone. Am I audible to everyone?
Yes.
Good evening, everyone, and a happy new year to everyone. On behalf of everyone in this group, I would like to take a minute to extend thanks who made today's event successful. First of all, I would like to thank today's speakers, Mrs. Shobha Methwani and Dr. Abhishek Mishra sir, for sharing their precious words and time with all of us. Being a math teacher, personally, I was looking forward to hear Shobha ma'am after knowing her love for mathematics. Thank you so much, Shobha ma'am, for sharing your experiences and work with all of us. Very good and interesting examples are taken to explain the examination pattern and NEP. It was really a fantastic session. I have no words to thank Abhishek sir as he spared time for today's talk. After hearing your work as Sanjeev sir introduced you with all of us, I was thinking about a program I have seen on Epic channel, how different animals are created by humans, not by nature. It fascinated me to know about hybrid animals and hybrid vegetables. And as you said, how gene editing will help in cure of different diseases. Thank you so much, sir, for sparing your time. I would like to thank Mrs. Priyanka Varshney, ma'am, for summarizing the speech of our speakers so effectively. I got a chance to hear Priyanka ji after a long time. I would like to thank members of Varshney Teachers Group who organized this webinar and Mr. Gagan Gupta sir for giving me the opportunity to present the vote of thanks. Lastly, I would like to thank all the participants for sparing their precious time and joining us today. Thank you so much.
Thank you. Namaskar.
Namaskar and good night.
Thank you. Namaste.